All right, great. Welcome everyone to our monthly variant type series here at VICC VMTB. And it's my great pleasure to have our colleagues here from the NCT in Heidelberg, Veronica Teleanu, and from the Technical University in Munich, Annika Schneider. And we're also waiting for Peter Horak from the National Center for Tumor Diseases. And these colleagues are working in a great project here in Germany, the MASTER trial, which is molecularly aided stratification for tumor eradication, which has been running since I think 2012 now, and it has provided great knowledge and methods, and we are hoping to hear how they actually now implementing proteome profiling in precision oncology, and I'm very much looking forward to the talk. The stage is all yours.
So, hello, and thank you to the organizers. And yeah, we are happy to be here and to share with you some of our experience of integrating broad molecular data that goes beyond genomic and transcriptomic. In this case, proteomics for clinical decision making in the frame of a molecular tumor board. And this is just a brief introduction to the programs, precision oncology programs run here at the NCT. So you can see there are, on one side, we have the routine molecular diagnostic. We run with colleagues from the pathology department, and that comprises gene panel sequencing, targeted gene expression profiling. And on the other hand, there is the more broad molecular analysis that is part of specialized precision oncology program, and that goes beyond routine clinical care. And that includes whole genome sequencing, whole exome sequencing, transcriptomic, and methylome. And for now almost 1 year, we also integrate proteomic data. And these programs are dedicated to different patient populations. So as you can see, there are master and inform programs that are actually cross-entity programs dedicated to young adults with rare cancer. That's the master program, and for children, that's the informed program. And there are also entity-focused programs that address breast. They are dedicated to women with breast cancer, that's catch and cognition, or to patients with brain tumors, this M2 program. And of course, can go to the next slide, please. These results are always discussed in the frame of molecular tumor boards. These are interdisciplinary session where colleagues from very different preclinical and clinical discipline participate. And currently we run 4 tumor boards per week here at the NCT Heidelberg. We have 2 panel tumor boards and the 2 boards, molecular tumor boards dedicated, as I said, to this more broad molecular analysis. So from the clinical perspective, go to the next slide, please. All these new techniques, they have limitation and they have strengths. And starting with the genomics that we use here since 2013, starting with whole exome, then going further to whole genome sequencing. It is the most used approach. We have the highest amount of data available and also the most robust clinical evidence. And we have clinical data that documented success of implemented therapy across a broad spectrum of malignancies, but there are also some limitations we encounter with this technology. So there are only some patients that harbor high actionable gene variants. Most very often cancer feature transit the genome that are not detected by this this technology, and it is also difficult to differentiate between drivers or bystander mutations. So I think we adding transcriptomic data to this layer is definitely increasing also the biomarker landscape and improved detections of some alteration that specifically gene fusions can be most reliably detected using RNA sequencing. Helps us validating some also others DNA alteration, and it's somehow also complementary use with metallomic profiling that gives us hints to the tumor of origin. But here we also encounter some limitation, and I think the most important limitation we see in daily practice is that there are less clinical evidence for RNA-based biomarkers. There are only a few clinical trials available up to now that use RNA expression-based selection criteria, and we often have the situation where we must validate the expression using other methods like immune histochemistry, for example. And moving to the next layer, so we are happy that we also can integrate phosphoproteomics data that helps us detect the protein levels that actually are also the direct targets of the most kinase inhibitors that we recommend. We can detect aberrant oncogenic signaling pathway activities. So we are not just restricted to one alteration. We can use this data, and Annika will show in the next slide how this data can be used to show actually to illustrate the pathway activities. And we can also validate the findings we have already detected using genomic and transcriptomic, but even more here with this technology than with transcriptomic. Here we also lack demonstration of clinical benefit, and the analysis of this data is still very complex, and I will give over to Annika, who will give you a more in-depth illustration of how we use and this data, and happy.
Okay, thank you. So as Veronica already said, I will just give a bit more detail on how we implemented the proteomic and phosphoproteomic workflow within the master and inform registry trial. And yes, this is actually for some well-known workflow. It's from patient enrollment towards personalized treatment recommendation. And at this stage here, we try to add proteomic and phosphoproteomic data to the genomic and transcriptomic information to complement and improve the personalized treatment recommendation. And this is now actually a rough overview of our workflow. And I won't go into detail. It's just some important information here. So we use as input material, we use fresh frozen tissue or lysate from the DNA and RNA extraction. And we multiplex 8 patients in one batch. And just a quick overview. So whenever we receive the samples, we extract the protein, digest the protein, we label the patient's peptides with stable isotopes, and then multiplex the patients, fractionate again to get a better coverage. And then we split up the sample, and the global proteome is measured afterwards, while for the phosphoproteome there is an extra phosphopeptide enrichment step before measurement. And both samples are afterwards measured via LC-MS system, and thereafter database search is performed, and we apply an in-house developed computational workflow to finally receive the patient MTB reports, which contain the protein and phosphosite expression data for each patient. And yeah, as I already said, this is now possible within 2 weeks. And to give you a quick overview on the cohort, so far we measured more than 300 samples within the master and infant program. This is mostly sarcoma and chordoma samples, and there are some or very few other entities in the background cohort at the moment. And there is a cutoff for the tumor cell content, so we include samples with more than 20% tumor cell content, so the tumor cell content scatters between 20 and 100%. And we're able to identify 8,000 proteins per patient, or more than 8,000 proteins per patient, and more than 20,000 phosphosites per patient. And this is actually quite stable. And just very brief on the reproducibility. So we process samples now for approximately 2 years. And this is just principal component analysis where you see all patient samples in green, while the QC channels, so I think I missed to say that in the workflow, within every batch there are 3 QC channels included, which are processed and measured in each batch. And they actually cluster nicely together. And also for the patient samples, which we just measured replicatives from over the 2 years, for some patients at least, you can see here that the replicates kind of cluster together. They're not, for some it's quite good, for some there is some difference, and those differences seem to be, or seem to result mostly from, yeah, there's a question, I guess. I saw a hand.
Yeah, sorry, you just said that you have patients replicates over the time, or is that from the same time point?
No, it's not longitudinal. It's just that if we have enough material per patient, we just process the patients twice, or already sometimes 3 times. It's actually the very same sample. It's just at this time, it's just about reproducibility.
Yeah, okay, so it's not about the change of the tumor over time.
No, it's not longitudinal. It's not a different material, right?
Okay. Thank you.
Yes, and yeah, just very brief. I mean, this is quite busy here. It's just to show that, I mean, we multiplex 8 patients in a batch, and these batch are just color labeled, and they have different forms. It's just to actually see that there is no pattern. So there is not really batch effects observed here. And now, just after the brief introduction, I just want to show how the analysis of those proteome profiles tend to look. And this is just some excerpt of a proteome profile from one patient. And there's always a long list of proteins given as gene names, and this list is actually 11,578 proteins long. And for each detected protein, there is we give the rank. So according to the intensity detected in the cohort, we give a rank the occurrence. That means how often is that protein detected in the cohort? Is it detected in each patient or just in a few? We give the fold change over the median of the cohort, and we z-score the intensity of the protein. And I color-coded this to just indicate that there's always, for each patient, there is overexpressed proteins in red. There's non-regulated proteins. This is just examples here colored in black. And there's underexpressed proteins, or even proteins that are not detected in that batch, in that patient. They are sometimes detected in a batch. And to simplify the analysis and to make it a bit easier, we just assign, or we just annotate the proteins that can be assigned to one or more clinical intervention baskets. So, and these baskets are mainly druggable signaling pathways, so like PI3K, KT-mTOR, MAPK-RAS. We have the tyrosine kinase signal transmission. There's immunotherapy, there's cell cycle, there is DNA damage repair, and so on. And we further look deeper in the receptor tyrosine kinase signaling, and we just also annotated proteins that are assigned to some receptor tyrosine kinase signaling. So like the receptor tyrosine kinase itself, and then the direct adapter proteins and the signaling proteins, which are sometimes unique and sometimes not. And yeah, at the moment we look at 960 proteins. This is less than 10% of what we detect, but this list is hopefully growing over time. And we apply the very same to the phosphoproteome profile.
Annika?
Yeah.
Looks like we have a question in the chat
I'm sorry.
about that. No, that's okay. Sorry for interrupting. We just want to, somebody asked, what is the rank based on? So how do you determine the rank in the last table? Or even in this table? Yeah.
Yeah, it's, I mean, it's same principle. So the rank is based on the detected intensity of the protein in the cohort. So if a protein ends up with a rank 1 in the cohort, it's the highest intensity we ever detected in the cohort.
And how comparable are these intensities, if I may chime in?
Yeah, there's, I mean, there's a lot of course, there's in the pre-processing pipeline, there is some normalization applied. So we just normalize within the batch and between the batches, but the correction factor is actually not too high. So it's based on the median intensity distribution of all detected peptides. Is that understandable?
Yeah,
Okay.
I think so. And so to follow up, if your cohort gets exceedingly large, is there a way to understand like what ranks mean, overexpression, and what ranks mean underexpression, or are your cohorts a pretty standard size?
No, at the moment we haven't stopped. So the background cohort is actually growing, and at some point we might stop. But within this 300 profile samples, there are more than 35 different histologic subtypes. So we had the idea that it really gets more stable if we profile more patients.
Yeah.
And yeah, I mean, overexpression, that is the thing that you will also notice when I follow up on the analysis. There is not really a hard cutoff value, because this also tends to, I mean, the tumor cell content is also important regarding the overexpression.
Right.
And it's a bit hard to say, okay, there's a cutoff for, I mean, yeah, of course, it's significant if it's the top 5 or 10% in the cohort. One can set that a hard cutoff, but at the moment we don't. So I will just show that. So we are to score on pathway activity, we just use the Z scores. And at the moment we do not only use the significant up or down regulations. Maybe that will get easier to understand if I show how we analyze that. Or is there another question?
Maybe one question also as a follow-up, because right now it's really dependent on what type of tumors you have in your cohort. As you said, 35 different histologies. Do you have any plans to include normals at some point for a comparison?
Yeah, I mean, it's actually really hard to get. I mean, at least to get a really healthy tissue. And it would be really tough to decide what tissue to take. I mean, those tumors, they tend to be located like everywhere. So it's not that there's one tissue of origin for those sarcoma. It's just very diverse. So I would say it's not. Yeah, Peter, you had a comment?
Sorry, I didn't want to interrupt, but I just wanted to stress what you just said, that we are looking at rare sarcoma subtypes, and there's most of them it's the same with the RNA expression analysis. We do not know what the cell of origin is. We do not know what the normal tissue should be. So there's basically no possibility to look at the normal cell for these rare sarcomas.
Yeah, it could have just been that if you have a biopsy where you could basically, I don't know, take certain areas which are more like to surrounding tissue.
I know what you mean, but of course, for liposarcoma, you could take fat tissue, but for Ewing sarcoma or for subsynovial sarcoma, there's basically no. And then, of course, we look at tissue-specific expression as well. So yeah, probably just looking at the expression as RNA and protein are highly tissue-specific. So we do not have a good baseline if we look into sarcomas and then take a, I don't know, muscle tissue, because it's not the same.
Yeah, it won't have the same profile. I get it. Cool.
Yeah, so I think I mean, eventually it's possible to do it for some patients, but I think it's really tough to base the analysis on it. Would be just interesting to know how it looks. But yeah, as it's not possible to do it for each patient, it's just the analysis is based on the background cohort, which is also, but the good thing about the diversity is that it should be possible to detect aberrant signaling, because it's just very diverse cancers. Yes. And yeah, I think I was just telling that the phosphoproteome, that the principle is the very same. So we just detect more than 100,000 modified sequences here. And they are always given. So there's the protein, the phosphosite is on, we give the modified sequence, there is the position of the detected site, and then there's again the rank, the occurrence, the fold change, the Z-score. And in case this site is annotated in phosphosite plus, we can give the responsible kinase or the responsible kinases. And in case it's known, there's also the effect on the modified protein given, and this information is just directly imported from phosphosite plus. And the proteins are assigned to intervention basket, same principle than with the global proteome. And yeah, just to give you here a rough numbers, so only 5% of the detected sites are annotated in phosphocyte plus, and yeah, 11% of the proteins are assigned to any intervention basket. And now just I just want to show how that in the end results in the detection of aberrant oncogenic signaling. And this is, as you saw already before, this is just now one example patient where we detected hyperactive PI3K/AKT/mTOR signaling, and this is just the proteome filtered on the PI3K/AKT/mTOR intervention basket, and I just I haven't shown all proteins, but it's here are the most relevant upregulations shown. There's some just to that you get the principle. There's some proteins shown that are not regulated, so the Z-score is around 0, and then there are proteins which tend to be downregulated or underexpressed in that tissue, and there's a negative Z-score here. And this is just now the core of the PI3K/AKT/mTOR signaling basket, and it's as you can see, it's just colored. There at the moment just two colors. It's red and blue, and it's just for the red colored protein. They this is just done literature based, and it's just done on the most evident that is available. But it's if these proteins are overexpressed, they either lead to pathway overactivity or they result from pathway overactivity. And for the blue colored ones, it's the other way around. If they are underexpressed or lost, or there's loss of function, these proteins lead or can lead to pathway overactivity or result from pathway overactivity. And this is how the scoring principle works. So the red colored proteins in the pathway, they have a positive weight. So if they're detected to be overexpressed, they score positive in the pathway activity score, while the blue colored ones, they have a negative weight. If they're underexpressed, they score positive to the activity score. And yeah, for this patient, it's just shown here. It results in a high score for the PI3K/AKT/mTOR basket. And this information is then overlaid by the phosphoproteome data, and this is again also filtered on the PI3K/AKT/mTOR basket, and it's also just some phosphocytes shown. It's just the most relevant upregulations, and I also filtered on the annotated one, so there's just the responsible kinases known and the effect on the protein. And yeah, you can see that there's for mTOR, there's a really high upregulation of an activity-inducing site, and there's mTOR targets highly phosphorylated. There's activity-inhibiting site on P10 and on TSC2. And in the end, this is just if one draws that in the pathway, one can see there is quite some activity-inducing phosphorylation within the pathway, and there's also on some mTOR targets or on some proteins inhibiting the pathway, there's also activity inhibiting phosphosites. And in the end, the PI3K/AKT/mTOR phosphoscore score for this patient was now detected to be high. And to just show how this can be useful is just if that is now a patient with there is high PI3K/AKT/mTOR activity observed, and in this patient we just on the first glimpse, if we see the data, one can see, okay, there's high a signal in the PI3K/AKT/mTOR baskets on phospho and full proteome. And there's also seems to be some upregulation in the MAPK pathway. And this is then in the end often manual analysis to check whether there is significant activation or whether this is just cross upregulations in other pathways. And but there are also profiles that look very different, and there's then significant scores in a lot of baskets. So here we see the tyrosine kinase basket score, and we see MAPK-RAS, PI3K/AKT/mTOR, and there's also, I missed the star, it's NF-kappa B high. And for the phospho, all those intervention baskets score high. And in this patient, there is really high PI3K/AKT/mTOR signaling, but there seems to be also signal in receptor tyrosine kinase baskets. These are, it's just important to differentiate on the first glimpse when you start to analyze patient to, while manually analyzing the profiles, to know where to look at. And yeah, now we just thought to show how we apply this. We just make some examples, and I think we do it as we do it in the tumor board. So Veronica, I think, presents the patient.
Yeah, we just some examples. Some of them we already presented in our molecular tumor boards, and there are cases that were analyzed prospectively. So we had all the data at the time of the molecular tumor board, and there are some cases later where we had the proteomic analysis after the patient was discussed in the molecular tumor board. So yeah, this is an example. This is a woman in the 50 years old with a glioblastoma. She underwent tumor resection and radiochemotherapy. She received over a long period temozolomide, underwent a second resection in progress, and was treated with multi-kinase inhibitor, regorafenib, followed by immune checkpoint inhibition with pembrolizumab. And after pembrolizumab treatment, she underwent another resection, and the sample was analyzed in the master program. And this slide shows the main findings from the genomic and transcriptomic data. So we had a quite high tumor mutation burden that was also explained by the typical temozolomide-associated mutational signature. From the other alteration we identified, we had a PTEN SNV, which we classified according to our VICC SOP as FUS, and I didn't show it here, but the expression of PTEN, RNA expression of PTEN was not that low. So this second another finding was, as I said, hypermutation. We had an amplification of 12q that is a logs for some genes that are recurrently amplified in glioblastoma, CDK4 and DM2. And probably the most important finding we also used for therapy recommendation was this highly EGFR amplification. And if you go to the next slide, we using also, it was actually a very complex structural variant, but using also the data from RNA sequencing, we could see that expression of exons 2 to 7 is very low and was actually pointing to this EGFR3 variant that we encounter in approximately 20% of the glioblastoma patient at that Randerson ligand independent constitutive activity of the receptor tyrosine kinase, and also with downstream activation. And Annika will take over for the proteomic data.
Yeah, actually, and we can also see that there is, so often we do also see high tumor mutational burden in terms of there's a lot of significant scores in those patients, and the profile of this patient immediately hints towards some receptor tyrosine kinase signaling, and this is just because there is significant scores in the tyrosine kinase basket. There is significant score in the receptor tyrosine kinase signal transmission. Then there is a significant score for PI3K, AKT, mTOR on global proteome and phosphoproteome, and for MAPK-RAS and FKB, and also Wind-Notch hedgehog signaling pathways on the phosphoproteome. And one checks down the receptor tyrosine kinase signaling basket, as I already introduced. We do see significant scores for not only EGFR, there's also some score for PDGF receptor, FGF receptor, and NTRK. And on the phospho, there's also additionally some other scoring. And this is what I actually just want to show. This is done in the end manual analysis to differentiate, because there's a lot of shared adapter proteins between receptor tyrosine kinase, and that is why the score is not always completely conclusive. The only thing that can be immediately said is that there is some receptor tyrosine kinase signaling. And if one checks down the pathways, and one then needs to do that for all the ones that scores, and here's just now the EGFR pathway shown, and this is the proteome profile. It's that one can clearly see here that there's EGFR and a lot of proteins within the EGFR signaling pathways highly upregulated, and also downstream in the MAPK-RAS and in the PI3K, AKT, mTOR basket, there's some upregulations. If one looks at the EGFR signaling pathway, it actually looks like this, and all the red proteins, they are overexpressed, not all really highly significant, but a lot of them highly significant, and the blue ones, they are underexpressed, while the ones in white, they are not regulated in that patient. And if one overlays this information with the phospho information, one can here immediately see that there is quite some phosphosites on EGFR itself, and there are two PY sites, so autophosphorylation sites, which are known to be activity inducing on EGFR itself. And there are also some others that are not annotated, but compared to a lot of other patient EGFRs here are highly phosphorylated. And this is also true for GAP1, for protein kinase C, for SOS1 and 2, SHP2, PLC gamma. So there's a lot of downstream adapter and signaling proteins phosphorylated. And yeah, this is just the pathway drawn with the phosphosites. So there's really high activity in this pathway observed. So, and the conclusion here from the proteome and phosphoproteome data is that there is a hyperactive EGFR signaling. And for this, and this is just now a summary of all the data, because there was not really, for us, no P10 loss or significant underexpression was observed. And yeah, this was now the most significant finding for that patient. And yeah.
Yeah, so the choice of the therapy, I think, was very well supported, but by this data, I think the problem we had, so we recommended EGFR directed therapy, and yeah, with a lower priority mTOR inhibition, and I think for the EGFR directed therapy in glioblastomas, there were several trials where different inhibitors were tested. The problem is also they do not cross well the brain-blood barrier, and they actually were mostly ineffective. But our choice of therapy was based off rather preclinical data and biologic rationale. There are some preclinical data from glioblastoma cells that showed that osimertinib reduced kinase activity and downstream signaling of cells carrying this variant, and also we know from lung cancer that osimertinib do crosses well the brain-blood barrier and is effective for cerebral metastasis, and the rationale to add bevacizumab based also from preclinical data that showed this also this glioblastoma cell lines carrying this EGFR variants impaired physiological adaptation to starvation, and adding bevacizumab renders the cell to apoptosis. So there are also clinical trials ongoing with this combination. So this was our therapy recommendation based on this data. Unfortunately, we don't have a follow-up, and we don't know if this therapy was implemented and how it worked. So
maybe there's one question before you continue with the next patient. There's the question whether you normalize the phosphor data to the full proteome data, or how are you doing the normalization of the phosphor data?
No, so the phosphor data, they're not normalized to the proteome data. And this is, was discussed like several times. And I think there are some approaches to do so, but there is actually some problems. So, there's not all proteins that are phosphorylated detected on for the full proteome, but this is just one thing. And the other thing is that it is really difficult to say that this always correlates. So that high phosphorylation it can come from high protein upregulation, but it's not only. So there's some structural phosphosites, and there are really some activating phosphosites. And if one would always normalize that to the protein level, I think one would really miss a lot. So I think it's important to know that the protein you observe the phosphosites on is highly upregulated, and that this might be a reason for some phosphosites being high. So we at the moment don't do that.
So if you actually then compare the different analysis, it's really based on the ranks in comparison to the cohort.
Yes.
Thank you.
Welcome.
So just a quick to the next case, medical history. This is also a young woman with a fibrosarcoma of the gluteal region. She underwent several surgical intervention and also chemotherapy, and was diagnosis was refined to dermatofibrosarcoma protuberans, also at the time when she was enrolled to the master program. So again, this slide shows you the main findings from the genomic and transcriptomic data. So we identified typical fusion for this entity. We had also highly expression of the PDGFR and B reception, and also some other tyrosine kinases, NTRK1 and FGFR1, and interestingly in the PI3K ACT-mTOR pathway we only detected an amplification of raptor and associated with RNA expression, and we had BAP1 focal homozygous loss, and from the MAPK basket we had high expression and amplification, focal amplification of crackle. Important for this patient, also for the therapy recommendation, and also the BRCA signature we had, and this patient would qualify also for a clinical trial here at the NCT that tests PARP inhibition. So that is not a focus of this presentation. So I think I will skip this part and hand over to Annika for the proteome analysis.
Yeah, so I mean, we decided to also show such patient to show that it is not always a very clear scoring principle, but so as you can see here, there's the scores for the tyrosine kinase and receptor tyrosine kinase signal transmission for the proteome, and there was absolutely no significant score on the phosphoproteome. And for the receptor tyrosine kinases, the PDGF receptor beta basket scored on full and less significant on phosphoproteome, and for the global proteome there was also EphREN basket scoring. And if we again do pathway analysis, so these are the upregulations in the PDGF receptor beta signaling pathways. So PDGF receptor beta itself really high, PDGF-B also highly overexpressed, but also not really detected in too many patients. And there, although there's not really significant score in the phosphoproteome, there were some phosphosites in the PDGF receptor beta pathway observed and on PDGF receptor beta itself, and there was also one autophosphorylation site. And so this is again an overview, but one of the bigger differences between those the first and this patient is that there's kind of strong PDGF receptor beta signaling itself observed, but there's not really downstream signal transmission observed to towards PI3K, AKT-mTOR or MAPK-RAS. And so the conclusion here is that there is hyperactive PDGF receptor beta signaling, but the note is that there's not strong downstream signal transmission here.
So our recommendation was kinase inhibition and with drug imatinib, and this was a retrospective case. So we had the recommendation before we saw the proteomic data, and this was based also on clinical data that are available from clinical trials that imatinib is efficient in this patient with dermatofibrosarcoma protuberans carrying this gene fusion. So yeah, I think this data supports our recommendation, and as I said, the other recommendation was based on the genomic and transcriptomic data, and yeah, proteomic data are not shown here. Yeah, this quick. This is another sarcoma, also in a very young patient with this was a high-grade sarcoma. Initially, she also underwent several local therapies and aggressive systemic therapy, and eventually progressed before she was enrolled in the master program. And according to the findings from the master sequencing, diagnosis was also refined to sclerosing epithelioid fibrosarcoma that due to this specific fusion. And beside this fusion, we also had in the kinase basket overexpression of FGF ligands, and also RTKs FGF R2 that we addressed therapeutically at that time. From the PI3K mTOR basket, we only had raptor overexpression, and what we found at that time, this is in some older case, was a SMARCB1 SNV, which we now in the light of the new also classification system categorized as a VUS. And I will hand over again to Annika for the data.
So and in this patient, the profile looked unexpectedly, so that we say there was not too many or too high tumor mutational burden, but there was a lot of significant scores here, and it from the scores already again immediately looked like receptor tyrosine kinase signaling. And also in the receptor tyrosine kinase signal baskets, there is quite some receptor tyrosine kinase basket scoring, and here it's done really manual analysis to try to find out which, or sometimes it's also just that, as we see next, not possible to really track it down to one receptor tyrosine kinase. But just to show that for the genomic data, there was only, or it was transcriptomic data, there was only raptor overexpression, and we actually also see the raptor overexpression, but there's actually quite a lot other proteins in the PI3K AKT mTOR pathway highly overexpressed, and there's also some downregulations or underexpressions here, and there's further several. I mean, that's actually the patient we had the example to show how the scoring works, and this really a lot of activating phosphosites in the PI3K AKT mTOR basket. And I mean, we saw that before. There's quite high PI3K AKT mTOR activity. However, it really seems that there's also receptor tyrosine kinase signaling. And here it's now really hard to track down. So there's some so ephrin A3, ephrin A4 upregulated on the global protein level. There is also some other less significant upregulations, but it's sometimes also that's what I meant with there's not really a hard cutoff here. And sometimes the phosphoproteome really helps to track it down. So in the phospho, we do see some KIT, some phosphosites on KIT. KIT was not overexpressed. We see phosphosites on ephrin A3, ephrin B2, and also some other receptor tyrosine or non-receptor tyrosine kinase. And there were also a lot of receptor tyrosine kinase signal transmission, so adapted proteins and signaling proteins highly overexpressed and also highly phosphorylated without showing all the phosphosites here. So so that's the conclusion from our data, is that there's really significant PI3K/AKT mTOR overactivity, but there's also tyrosine kinase signaling, which is definitely hard to track down to which receptor tyrosine kinase. There's some, it's possible that it's KIT or FRNA3, but it's not really it's not possible to define that really clear. So
yeah, so this case was actually interesting because our initial recommendation is retrospective analysis with proteomic data, and our initial recommendation we're going on some other pathways. We consider actually SMARC B1 loss as a potential target and recommended tazemetostat, and of course the overexpression of some RTKs to recommend tyrosine kinase inhibition. And we didn't recommend mTOR inhibition based on the findings we had from genomic and transcriptomic data, so there was not really strong clues towards mTOR pathway activation. And considering now the whole data, I think we would have chosen mTOR inhibition as the first option, and probably it would make sense to combine this to target both pathways with a tyrosine kinase inhibitor and mTOR inhibitor. So and this is our last case. This is also a very interesting case.
Just to let you know, so it's now 6 p.m. People might be leaving. The question is whether we want to do a quick discussion round now or look at the patient. I don't know, Beth, what do you think? So I find it really fascinating, and the question is whether we can also have some discussion with the people who might have to leave.
Sure. I think that if anybody has any questions, just post it in the chat. Even if you do have to leave, we can send those forward to our presenters and they can get back to you. But let's continue on with the recording and the presentation, and then we'll make the rest of the recording available on our YouTube channel today. I'll send out the link in an email to the VICC distribution list in case people have to leave. You can catch this last case. Does that sound okay?
Yeah,
I think so.
yes, it's
Perfect.
fine.
Yeah, we started a couple minutes late, so I don't mind going a couple minutes over with this. Yeah.
Okay, perfect. Yeah, then please continue, and sorry for interrupting.
This is the last patient. It's a young patient with a round cell sarcoma with an EVSR non-ETS fusion that subsequently was also better classified as this very seldom fusion. That's one. He was treated with intensive chemotherapy and progressed after 5 cycles, and afterwards enrolled in the master program. And this is very surprising finding. Unfortunately, due to quality problems, we didn't have transcriptomic data in this case, but it was possible to detect the fusion. And the main and unexpected finding was this BRAF mutation. This is a class 3 mutation. I just made an extract from Cancer Hotspot to illustrate how seldom this mutation occurs comparing to the most frequent hotspot V600E mutation. And yeah, this class 3 mutation, they also behave differently than other this yeah, class 1 mutation, and that this is a kinase dead mutation that activates the MAPK signaling by dimerization. It has a high affinity to CRAF, and this drives the downstream pathway activation. And yeah, the therapeutic approach for this mutation is also different. And yeah, the proteomic data gave us a good insight into this.
Yeah, so I can make it very quick. So, I mean, this is that's how the scores look like. And as we see, there's quite a really low score on MAPK RAS on the global proteome, while there's a high score on the phosphoproteome. And there's actually nothing else scoring in the receptor tyrosine kinase baskets. And if one actually looks at the proteome to find out why the score is actually really low, it is that there's only RAF being highly overexpressed, so CRAF, while BRAF is down, and actually all detected RAS variants are very low expressed or underexpressed, and there's slight overexpression of ERK2 and MEK2, but there's also underexpression here. And this is how it looks. But if one checks the phosphoproteome, one can really see that there's a lot of phosphorylations on RAF1, either activity inhibiting or activity inducing. And there's also a lot of phosphosites on MEK and on ERK observed. And I just didn't put the long list, but there's quite high phosphorylation of ERK substrates. And yeah, so the conclusion is here that we for sure observe MAPK hyperactivity on the phosphoprotein and phosphoproteome, and the profile on the global proteome is actually just explainable by the findings for this specific class 3 mutation. And yeah, this is now.
Right, so we supported also by this data, we recommend a pan-RAF inhibition, and fortunately this is also possible within a clinical trial that is enrolling patients also here in 2 centers in Germany. And the idea stemmed also from a patient with melanoma carrying this mutation who responded to the combination of sorafenib and trametinib. And yeah, there is also another option to treat the patient also within a clinical trial with a pan-RAF inhibitor, belvarafenib. And yeah, we hope this is as a prospective case, and yeah, we are really excited to see if the patient will benefit from this one of those therapeutic approaches.
Yes, and this is now due to the sake of time, it's very quick. It's just that there is we established a workflow that is working in a 2-week turnaround time to do phosphoproteome and proteome profiling. We profiled more than 300 sarcoma and chordoma patients. We developed an approach to infer aberrant activity of druggable signaling pathways using those expression data. And initial data really support the idea that adding a proteomic layer can support and add critical information for molecular tumor board treatment recommendation. However, of course, as we just started with prospective samples, it's really hard to tell whether there's any clinical evidence. So, and the outlook of this whole project is that we plan to integrate proteomics into prospective molecularly informed clinical trials to improve clinical evidence and molecular stratification, and hopefully get some clinical evidence in the future. And with this, I think there's just a lot of people we have to thank. It's a huge team working on the project. And yeah, thank you for the attention. And if there's some time, we are happy to answer some questions.
First of all, thank you for this great presentation. That was very intriguing and informative. I think we have a couple of minutes for quick questions. Any questions from the audience? I don't see any hands raised right now. Maybe I have one, and then people can also write their questions in the chat in the meantime. So you showed like 4 cases, and in some cases you actually saw that adding proteomics data would change the recommendations in the molecular tumor board. How often did that happen for the 300 some patients you had now?
I mean, I don't know if I need to answer, but so we haven't discussed all 300 patients in the tumor board. We discussed, I think, retrospectively around 80 and prospectively now around 25, if I'm right. And I think we haven't done the whole meta-analysis. If I would have to guess, so we kind of really sometimes change the prioritization, so the order in some cases. So if I would need to think in percentage, it's maybe 40%, but I really can't tell a number. Yeah, and then in some cases we just saw some things in addition that would be nice to note down to see whether this in the end further stratified the patient towards a treatment response. But I think, yeah, I mean, we really need to track down the, yeah, or would you say something different, Veronica, Peter? I don't know.
Yeah, I think I agree. I also don't have exact data, but I think it's mostly for the receptor tyrosine kinase pathway and those recommendations. I think that proteomic data can add substantially to better guide us if this pathway is really activated, and which inhibitor we would choose, because this is also the level where really clinical evidence is somehow missing, and we are often in the preclinical or most biological rational evidence level. So I think this is a great benefit. And also, as the case and we had previously, where we have some data that are not very clean, pointing towards a pathway activation, as for mTOR, some copy number variation, some amplification, but not that clear that we would really recommend a clear mTOR inhibition. And looking at the proteomics data, this might also change the situation.
Yeah, I agree that it's of course difficult as you don't have as much evidence for this kind of data. But it was interesting to see that you do have, like, that it actually gives you an additional layer for some of the cases. So I'm very interested to see how that evolves over the next months and years. We do have a question from the chat now. So usually we do not consider variants of unknown significance from genomic analysis. Do proteomics data help with functionally annotating them, and might that influence therapeutic decisions? Are you looking into that type of detail?
Maybe I can answer that. Yes, and I think that's one of the reasons for doing RNA and proteomic profiling. As you could see in one of these examples where we had a SMART V1 VUS basically retrospectively. Back then we didn't categorize it as a VUS. Probably with the additional data, we could have some or less incentives to recommend EZH2 inhibition. Yes, I would say that given that, for example, tyrosine kinase activating mutations usually lead to protein phosphorylation and downstream activity, we would hope to see some of the effect in a VUS, and of course it would help us categorize VUS is better without maybe before doing an in vitro study or prompting an in vitro study if we see an activating VUS.
Yeah, and I mean, we had already some cases where there's really a lot of receptor tyrosine kinases overexpressed, but there's absolutely no downstream signaling observed. And of course, it's now not super clear because the follow-ups on those cases are just not there. But if one can get these data somehow in the future, I think it would be an important information whether these amplification, in which cases the amplification leads to really signaling pathway activity, and what that means regarding sensitivity towards kinase inhibition. So this is a further possible application here.
Yeah, okay, thanks. Are there any further questions? If not, I think people can probably reach out to you if they have questions coming up later on as well. Thanks so much for joining. It was really awesome. We are looking forward to the next variant type series, which is on November 8th on HRD profiling. And I hope you all join us there. And yeah, thanks to Veronica, Annika, and Peter for coming and presenting their proteomics data today.
Thank you for the invitation.
Thank you.
Thank you so much.
Bye-bye.
Bye.
Bye.
Bye.